for introducing, you know, with enthusiasm the question of where does strategy take us. And obviously, we do want everyone's input on this. So this year, we really want to do this early because we've seen that the success of these contests really depends on getting the data out fast, and we want you to be a driver in this rather than just being told what we're doing. This year, we've had 3 challenges: the MetaSub Intercity Challenge, the Neuroblastoma Data Integration Challenge, and the Oxford Nanopore Wiggle Space Challenge. And the question is, how do we move from this? So these are a number of data sets that we will briefly discuss in this session and ask for your input. Maybe you have completely different data sets in mind that you think we should be looking at, in which case we'd love to hear from you. So Wenzhong has just discussed the question of looking into cross-species multiomics, not to figure out how rabbits are different from us, but to figure out why certain disease processes work so different in species that are, I mean, come on, we're all mammals, right? Chronic fatigue syndrome as a possible area where we could look into multi-omics data integration. And I will say a little bit with slides that we received from Wei Tong from the FDA about a possible liver injury challenge data set. But before I do that, I would like to invite Chris Mason, who has been the leading light on all things MetaSub, to come out and tell us a little bit more about how this newly forged alliance between CAMDA and MetaSub could be taken further in the coming years.
Great, thanks.
Okay, I'll show this video. But you can take a mic and start talking.
So good afternoon, everyone, or good evening almost. A lot of you looked at some of the data sets, or if you were at the talks this morning, about ways to look at large-scale metagenomics data sets, of which there were 3 that were released later last year, but we're now releasing many more. And I want to just play this quick video, which is a summary, including some dramatic music, of what the goals of the consortium are and what's coming from it. Okay, so very dramatic. Now you all feel like you're in a hypnotic daze right now, like you're floating in space. That's part of the idea. So this is, really something that is born out of, we talked earlier this morning about a lot of curiosity and excitement that we can now map a really invisible world around us. And there has been already a little bit of a love exchange between CAMDA and Metasub, and I think it provides a really great opportunity for complex and interesting data sets that also have metadata and geospatial information, and then allowing people to mine the data, come at the data with very different looks, different backgrounds, different ideas. Of course, there is eventually, some forensic and eventually clinical applications. We at Weill Cornell, we want to do this for not only cancer patients, but for all infectious disease patients. So these methods and tools and algorithms are not just academic exercises. There are ways to improve how well and how quickly we can diagnose and characterize any microbiome or metagenomic samples. So, let me go back to the slides. That's just my quick note about that's the background. Also, all of this is up on the. I know this is one things. All this is up on the website. No, where did your talk go? Is it PDF? Got it. Okay. So that video, if you want to watch it as you go to sleep tonight and just have the music play, you can do it there. But the video is online, and as it said at the end of the video, we want to make all the methods, the tools, the algorithms, and the raw data freely available, publicly available, but under a sort of a Fort Lauderdale agreement. So we'll release the data publicly and within the consortium, but also not want to publish it in waves and in concert with each other. So I just came back from the Faculty of 1000 group where they have the Gates Open Access research, and since this is a Gates Foundation funded research, that's one place where you can publish. There's also Biology Direct, which is kind of through CAMDA, so you really have at least two options for really quick papers that you want to get out fast, and CAMDA and Biology Direct are official partners, I think is the official collaboration, but also there is, what will be launched in October is Gates Open Research, which was similar to with some of the other open access journals, one that was done also for some of the HHMI kind of investigators, kind of like eLife. So what we're going to be doing next year and releasing in Chicago, the sequencing is ongoing. Most of the sampling has been completed for this year. There is some additional seasonal sampling that we won't be talking about this year, but we will have we have at least 10 cities sequenced. We should have probably, by the time we get done sequencing in September, probably about 30 cities that have been profiled and sequenced, potentially more, that will release the data publicly. This will include raw sequence files as well as de novo assembly. And the goal will be to have people mine it in general, so that much like what we saw this year, where you can mine it for specific phages, CRISPR elements, antibiotic resistance, human alleles, plant diversity, fungal diversity. If you're a viral junkie, you can be a viral junkie here. If you like more bacteria, human allele tracking is also going to be done by some collaborators. So there's a lot of complexity in the data that can be mined. And that'll be released essentially at the end of this year with CAMDA. But then also there'll be some mystery samples where you don't know which city they're from. We're going to see which kingdom of life might reveal the most about the provenance of a sample, what city. So it's a forensics challenge, a metagenomics forensic challenge that we will have next year. So there are some samples that will be blinded, only maybe a few of us will know where they're from, so we won't be able to contribute. I think even I won't know where they're from, maybe just to keep it very fair. I will have no idea. There'll be a very small group of people that know the answer, and we'll release these as mystery samples to see based on, say, K-mer profiles, species identification, different other, mapping to other species. Can you figure out what city the sample came from, which is an ongoing question we have in the consortium. There'll also be some of the same questions you can ask again about, types of biology or antibiotic resistance, which are the main foci of the consortium. Then there'll be a new Oxford Nanopore data set we have from clinical samples that we should release as well, and then. And I think you've discussed some of the other ones. Do you want me to jump to the next slide, or do you want to jump in?
Yeah, why don't you do the Metasub thing for 2019?
Okay, 2019. So yes, we have an idea for. So this is for Chicago for next year. The other good thing is there's a lot of metagenomics that gets done. Jack Gilbert's there. A lot of other researchers in the University of Chicago are not that far from where the meeting will be next year, so I think it should be, timing-wise and location-wise, a lot of great research that can come up. There's a microbiome center that's in Chicago, and also some work in Minneapolis and Wisconsin that's also very close, so it should be a good data set. And then, so we do have some samples that are from space that we will have likely released later this year, but definitely by the year we get to 2019. So it's not quite intergalactic metagenomics, but we will have suborbital metagenomic samples. So one question we'll have, and again, these are data sets that we have in-house, but are not yet published, and due to privacy concerns with NASA, we can't have this as a challenge yet, but almost certainly by a year and a half from now, we will have the raw sequences posted and then likely available through GeneLab, which is NASA's website of somewhat questionable utility generally, but at least it's their website for data release. It's criticized by everyone. They can never make anyone happy, but at least it'll go there where you can actually have samples from the space station. We'll also have astronauts who have come from Earth, spend time on the space station, and come back down. And there's an ongoing question to see, can we see what they picked up in their skin while they were living in a tin can, and see if you could predict that, because we'll have samples from the walls and what they were eating as well as their skin and their mouth, so you can see if you can pick it up. It's an interesting challenge for 2019. And then we'll also just have more cities that have been sequenced, and we'll also have RNA. So we'll get into metatranscriptomics data. RNA viruses will come out next year, so this will be something else that we can add to the challenge for the subsequent years. And with the same goal to release data, allow people to mine the data. If you have an interesting result that sometimes would end up as a supplement or a small paper, there's easy mechanisms to publish it with Gates Open Access or Biology Direct, and also some other projects that maybe David or Paul you want to talk about. But that's and then we won't go. It's more intergalactic was a pretty big jump. We should have started with interplanetary, I guess, or something. But we're not going to be intergalactic yet. Thanks.
Thank you so much, Chris. So as you can tell, anonymizing astronauts is rather hard. So coming back to more earthly challenges, obviously, convert? No, I don't want to convert anything. Yeah, so coming back to next year, exactly this question of whether we can get access to the data due to questions of patient consent, and so it puts a little question mark behind whether we can get access to the chronic fatigue syndrome data, including the genome, the DNA. But we'll see what we can do. Wentong is kindly pursuing that. Similarly, the situation is not entirely sorted for the cross-species multi-omics, but we hope that we can either get something for next year or at least certainly for the year after that. I want to say a little bit about the Fda data set, if I manage to copy this on here. Is there an additional slot that I can use? Or
Sorry, this is...
I need to take that out. Okay. Right. Okay, so these are slides by Fda Tong, and I'm going to whiz through them a little bit because I want you to listen less and to speak more. Basically, the idea is that this will be a hybrid CAMDA FDA gig, where basically we will be combining two different ways of getting the community engaged. One will be the usual open-ended data analysis contest that CAMDA does, where we're basically saying, here's a big set of data with interesting questions, see what you can do with it. And the other one that, because the FDA is a regulatory agency, so they need to be in control of some process, so they will offer a more streamlined, tighter, more directed way of looking at the data, where they basically say, you look at the data, you finalize your code at a certain date, you send them your predictions and the lockdown code, then they will send you a new independent data set that no one has seen before, on which you will be tested again. Then they will tell you the results, and then you can write your paper for CAMDA. So this is basically, you will have the strict approach where you get the benefit from an additional independent data set to validate your work, and you will get the open-ended, you can just look at whatever you like approach, and we will run those two in parallel. And the idea here is that most drugs that we try to develop fail because of unexpected liver toxicity. So this is a huge practical problem, but it's also from a theoretical point of view very interesting. Why can we not predict, or why is it so hard to predict whether a certain chemical substance damages liver? So this is the challenge here, and there will be two data sources that we will employ. One will be based on the chemical structure of the drugs and additional characterizations, and the others will be a genomics data set that's already a little bit older. And one thing in the open contest would be to just compare these, the in silico models based on the structure and the genomics-based predictions. The reason the FDA is trying to push this out in such a clean and controlled way is that they have found that the performance of an algorithm often depends on basically the entire setup of a validation, of a benchmark. So how large the data are, what type of endpoints you pick, what the distribution of bad drugs and good drugs is, so whether you have a lot of damage or a little damage. So that all determines which type of predictors actually work better. And so that you basically can assess this across, they will have a number of data sets, 5 data sets, where they have firstly different sizes, secondly different distributions of really bad drugs and middle bad drugs and not so bad drugs, but also different ways of classifying the drugs, so different ways of giving scores as to how badly a liver is damaged. Because for me as a data scientist, I just expect a score, from 0 to 1 that tells me how bad it is, but how do you get to that score? Obviously, there are different ways you can get to this score. So in the first data set alone, there will be 3 different types of score that summarize how badly the liver is damaged. And in data set 2, there will be 3 different types of scores, right? And so on. So you will basically get to see whether your approach is flexible enough to model liver damage in these different scenarios. And the toxicogenomics data is actually something that a lot of you may already be familiar with, because it's featured at CAMDA already for 3 years. In the past, it was a very popular data set. It's a large data set from Japan that includes expression data from Affymetrix arrays for human in vitro, for rat in vitro and rat in vivo assays for different doses and different dose schedules over time. So this is a very large data set that we can employ. So this is a little bit of the formalities. I will spare you this. I'll just leave it on the screen so that you will have it in the video that will be recorded by ISMB that you can check later. Basically, this is the process of the formal setup. If you go the Fda assessment route, here are the timelines for this. So the data will be released fairly early, already in September. Normally, we aim to release CAMDA data around November, December, so that you have about half a year until May to analyze it. So here, the fact that FDA wants to lock down your algorithm for a true double-blind assessment, the data will be released earlier in September. The lockdown will be then in February, which also gives you about five months. You will get the independent test sets, the validation sets, a month later, which you can then apply for a month, and then you still have about 1.5 months to write your extended abstract for CAMDA. The conference again will be in July, and publications will go out in September. As now, like anyone who's presented here is invited to submit a full-length research article to the CAMDA proceedings in Biology Direct by 24th of September. This will be exactly the same here, with the only additional difference that the FDA will have a manuscript lead where they will write a comparative paper about all the people that formally submitted their method to them in one meta-analysis comparison paper that they will aim to get in a high-profile journal where everyone who takes part will be a co-author. Yeah, but this solves the dilemma of what do you get out of participating in a contest like that if then there's one paper coming out, you're author number 17, and your method is number 7, right? That's not that exciting. So we split this up. You can have your own analysis first, publish it separately, and after that there will be a meta-analysis survey paper by the FDA. So we hope that that makes everyone happy. Yeah, so that's more or less the setup for the drug-induced liver injury. So let me get back to where I was before. Yep. So what we're also exploring to make the open-ended data analysis context for this data set more interesting, where we will have all these drug chemical structures plus the old data from the Japanese toxicogenomics project. We're also seeing whether we could get recent data from the broad, where they've done a connectivity map, where they've looked at tens of thousands of compounds in 100 cell lines, both with their L1000 system, where they have this idea that you don't need to measure all 50,000 genes to understand what's going on in an organism. It's enough to measure 1,000 representative ones. That hasn't worked so well in the past, so they're attempting this again. And they also have regular RNA-seq data for a reasonable subset of these drug effects that they're trying to measure. So no matter whether you think the L1000 are valuable in their own right or not, there should be additional data coming out there, and this is being published as we speak, so hopefully it will be ready in a couple of months. So that's the idea around drug-induced liver injury. Wensheng has introduced the other two. I will just skip ahead to 2019 briefly, and then I would like to open the floor for discussion, which Paul is going to moderate. So we already know that Metasub is going for the stars. By 2019, we should definitely also have these two other data sets. And there's also a possibility that we're very excited about, that through Jilyushi we will get access to data from the Chinese Human Proteomics Project, which includes clinical data and phosphoproteomic data, and will be accessible really from the raw data up, in contrast to a situation that we have had with TCGA, where you only get processed data. So that's it for me as an introduction and as an inspiration for what type of questions we could be looking at in the couple of years ahead. And I would very much like to invite all of you for comments, questions, suggestions, other ideas, other data sets, criticisms, whatever. Yeah, Pavel, please, do you want to organize this with people coming up here, or shall we just discuss in the round?
10 minutes.
Huh?
10 minutes.
We have 10 minutes. Well, then let's leave it open like that. Yeah.
So thank you, David. So now the question is, as we have asked you yesterday, to think about whether you would have any kind of the ideas and data set. So if anybody of you have questions to these challenges, or maybe have an idea about the new challenges, then please raise your hand and just pop by to the microphone. No volunteers?
Everyone at the same time.
Okay, Sergey.
One idea would be kind of to put the question and then let people, one idea would be kind of to phrase the question and then let people explore the data and find the data. Like, for example, you have the whole SRA of like RNA-seq data across different like tissues, diseases and stuff like that. So, and then kind of, like motivate people and then see who can answer with certain amount of data and see if getting more data will get you the precision. Well, so kind of do the Wild West in a way. You give people the question and then you tell them, hey, go ahead and explore all available data across the SRA or other databases and see how people can do. I am for, yeah, I need to think a little bit.
Yeah, it's interesting approach, but you say that we will need to ask specific question, right?
Yeah, I guess like you want to compare how everybody does.
Because so far the idea of the CAMDA was the other way around, that we're providing the data,
yes.
We suggest the route, but we are not close for this only question. You could see that we always are providing data, then we giving 2-3 questions, but these are not the only question which is supposed to be answered. If you will find something also interesting, then it's fine. So yeah, giving the one question, I'm not sure whether the...
Well, it would basically shift the flexibility the other way around. It would say, here's an interesting question, and you can bring different data to the question.
Yeah.
So far, we've had the data, and you can bring different questions to the data. I mean, either way, it might be interesting. I think it will depend on whether we can find a question that is both interesting and concrete enough, but still open enough. And there, I would very much look forward, if you had something in mind, Sergey, or anyone else, please do contact us. So the contest data sets will be finalized in the next 2 months or so.
Yeah, I just would like, before Chris will ask the question, it's interesting, but when we provide the data, we are giving what is, for most scientists, are missing, that many people who are developing new methods, they have ideas, but they do not have data to validate it. In case, when you ask the question, Of course, in the better position will be the person who has good access to the big data to answer the questions, and all the others, how you make sure that it will be, you know, a fair competition. So yes, it's interesting. If you have question in mind, then we can really consider it for next challenges.
I guess I'll just answer you. Blinding some of the data sets, or you know, you have to make sure they're blinded in advance. But I had a different question though. I don't know about the. To switch gears, one other thing, Lin Ming and I were just discussing some of the other data sets coming from the SCQC2 will be also the epigenomics data set. So there'll be also some basically sequencing, MinION and PacBio sequencing of the same quartet, the Chinese quartet. So it'll be interesting ability to look at validation of epigenetic marks in DNA, but then also we'll be doing some of the direct RNA sequencing. So those kits have just been released, but we've seen really interesting data so far for being able to sequence RNA directly. So not cDNA sequencing, which is the majority of RNA sequencing, but actually direct RNA sequencing. And also Lin Ming has already sequenced them with ribodepletion kits and some other methods. So we can compare. This came up in the original SCQC as a full length sequencing of really, you know, comparing the isoform distribution and samples. So I think we can revisit one of the questions from actually like 3 years ago is to have a really deeply sequenced and direct RNA sequence data set.
That sounds great. Would that be for time release more towards 2018 or 2019?
It depends on the next. In principle, it could be 2018. It depends on how the next 6 to 9 months go. But I think it
6 to 9 months, then it would be 2019 because people need to get the data to analyze them for about half a year.
We were hoping to be done by September, October, but given
this year,
but given.
But I think in general, any kind of the data with the long reads are opening a new venue for challenges, right? Because at the moment we don't have the big set of the data with the long reads. Once we have them, we can start answering many interesting questions which we cannot re-answer now.
Right, which we've been talking about for years at this point, right? In some cases, decades. Some people in this room.
This was the idea with the Oxford Nanopore challenge, right?
Yeah. So that's just something we were just chatting about. So that'll come out soon as well. And that could even be. Yeah, it's an old question that I think it could last for several years because it's there's other platforms coming out soon as well this fall, and some that'll be announced at AGBT. So it's a long debate of short reads, long reads, and we could even have like a wrestling match maybe on stage. I think.
Yeah. It seems to. Okay. Any other ideas? Or maybe you have comments to these challenges which were presented by Chris Wensong? Yeah, Cesare?
Do you have a map of results along the different challenges, how things are comparing each other, if there is a link between proposals made along a challenge and another?
Can you speak a bit louder? I cannot hear.
Oh, really? Okay, I was asking if have you been tracking results along challenges in the last years so that you can compare if some ideas used in the last 2 or 3 sessions can be combined and reused, or there is some line of thought, or maybe there's something happening and you can create a bridge between 2 ideas. So you have a marketplace of ideas and try to connect them.
Normally, if we use the same data set at least for 1-2 years in a row, depending what is the interest, we were like having this Oxford Nanopore already second year because it seems to be very interesting. But last year we haven't received any submissions, so we thought that it's not interesting. But there were people saying that it is interesting, that it's just hard. And this year we got just 1 submission. So the question is whether we should block, or because there's not always, we cannot predict how popular will be specific challenge, and there's not always possibility to compare between different years.
I was not really looking for, say, a competition, but possibly having sort of a list of methods, approaches. So yesterday we had a number of alternatives for the same data set, and wonder what else is happening for the MetaSub data. So I think it would make sense to start thinking how to compare ideas, methods, and possibly results.
Okay, yeah. So it sounds interesting, and we for sure have to consider it. Thank you. Okay. Yeah?
No.
So I think that as it seems that there's no more questions.
Okay, and you got the things for Mate, and is he around? Because now it's time for ceremony, right?
Yeah.
Okay, so firstly, to to wrap up, this is actually something, this idea that Cesare just raised is actually something that we do encourage everyone here to think about, because the research paper that you're submitting to the proceedings does not necessarily have to be identical to what you've presented here. So if from listening to other people's approaches and other people's way of doing things, you think you can improve your own work, by all means do. Yeah, so that's the whole point why we have this open exchange here, and why we don't just take whatever you sent us as extended abstract and dump it into a journal. We want you to basically do the best research that you can based on your exchange here. You can upgrade and improve your paper and then send it to us in September. Yeah. So that said, I still have the pleasant duty to inform you of the results of the Best Presentation Award voting. We had 33 people voting. Each person could hand out between 3 and 6 voting points. People who voted for the same paper multiple times, no, they weren't deleted, but I only counted their vote once. So with that said, I would like to start with the third prize, which includes a certificate and sponsored by F1000, and the ability to submit one article free of charge to the F1000 Research Journal. And this goes to Chandrima Bhattacharya. Welcome to CAMDA, and I hope you will come back often.
Yeah, I do intend to come back in the future year. This was my first time, and then as an undergraduate, it is student, it is really honorable to get this award.
And you get a prize. Brilliant. There you go.
Thank you. Thanks a lot. I would like to thank both of you for giving me the registration waiver, because of which I could attend the conference.
Very good. Pleasure. Thank you. The second prize, the second place, goes to the presentation by Moreno Solfo. This is also a free submission to F1000 Research and the certificate. Congratulations.
Thank you very much.
So the first place goes to the presentation by Mr. Mathieu Kanua. And that, in addition to the F1000 research publication, also includes a one-year membership to F1000 Prime, and we thank our sponsor, F1000. Mathieu, I'm pleased to award this prize to you.
Thank you. Thank you very much. Thank you.
And wait a second, not so fast. And as is tradition, the first prize at CAMDA also takes home the CAMDA trophy, which is always something that is reflective of our host country, and we're very grateful to Mathieu Lexa to help us find something impressive and fantastic. In this case, this is something based on garnets, red garnets. These are semi-precious stones, and they're actually embedded in the globe of science here. So if you want to have a look, this is for you to take home. And if you're taking the train, you're safe. If you're flying, don't take it in your hand luggage. Yes. So that's this year's trophy. Congratulations.
Thank you. Thank you.
And with that said, we also would like to thank our local organizer, Matej Lexa. We have a little something from Austria, which is where we're from. And we would like to thank you so much for being such a brilliant host and setting everything up.
Thank you very much. The pleasures are so mine.
So this is an ice wine because we do wine, but we also have snow in Austria. So anyway, so with that, thank you all for attending and for contributing. We much look forward to hearing from you in the next coming weeks. If you have any further ideas regarding the contests that come up, and we look forward to receiving your submissions for the Biology Direct CAMDA proceedings issue by the 24th of September. And I hope you enjoy the rest of ISMB.